The sketch shows the shape of a dissociation curve. Okay, we'll go back to our notes. And here we have the oxygen dissociation curve. If hemoglobin, if the oxygen binding with hemoglobin is happening just like a normal, simple chemical reaction, we expect the as saturation of oxygen with hemoglobin, saturation of hemoglobin with oxygen to be linear, it, to be a linear positive correlation with the partial pressure of oxygen as this predicted straight line. But what actually we have is the sigmoid curve with a positive correlation. This is due to the action of the peripheral protein component in the hemoglobin molecule, which is not acting as expected with a normal inorganic molecule. Let's see the structure of heme in hemoglobin. We know already hemoglobin is made of heme groups and globular protein groups. So heme group is consisting of this porphyrin ring, and in the center where you have an iron which is in a coordination complex with nitrogens in the porphyrin ring, 4 nitrogens from the porphyrin ring, and 1 nitrogen from the proximal histidine, which is a part of the globin protein. Still, there is a vacancy for another coordination bond which occurs with oxygen, molecular oxygen O2, and that is stabilized by a distal histidine as shown here, which is another again a, a part, an end from the protein component, globular protein component surrounding the heme group. This is how the oxygen transportation happens. Oxygen is in a coordination bond with iron, but carbon dioxide is binding with the protein group, amine group of protein. That is why carbon dioxide transported in hemoglobin is called carb aminohemoglobin, and oxygen, when oxygen binds with the hemoglobin, we call it oxyhemoglobin because it is not involving with any parts of the protein group directly. It is directly binding with a coordination complex mechanism with the central iron atom. Iron. Okay. So because of this, the binding capacity of oxygen, binding or affinity towards oxygen of this iron is changing with the behavior of these peripheral groups, the proximal histidine, distal histidine, porphyrin ring, and the conformation of the protein. Therefore, we get a sigmoid curve, not the linear curve as expected. So that's from theory. We'll go back to the question, which describes this dissociation curve. Simply, you can, even if you don't know this theories, all these theories and chemistry behind, you can easily see that this is a sigmoid curve showing a positive correlation. So the answer should be D. The next question, which name is given to the effect of increasing carbon dioxide concentration on this dissociation curve? Back to notes. So most of the, some of the carbon dioxide is transported in the form of carb aminohemoglobin. Some of the carbon dioxide is transported in the plasma. Some of the carbon dioxide is transported in the cytoplasm of the red blood cells in the form of hydrogen carbonate ions. Okay, let's see what happens when carbon dioxide binds with hemoglobin. In high carbon dioxide environments, carbon dioxide binds with the protein component of the hemoglobin and changes the conformation of protein part, and it causes the affinity of iron to be lower towards oxygen. This causes more oxygen to be released. This is an important phenomenon when it comes to respiring tissues. It efficiently gives out oxygen. In respiring tissues, we know that there is a high concentration of carbon dioxide. Therefore, there is a high possibility of carbon dioxide binding with the protein groups and releasing more and more oxygen into the tissue, which is very important to keep up the demand of oxygen of the tissue. And this effect, simply the question is asking what is the name of this effect. So that is Bow effect. Going further, explain how the structure of hemoglobin is related to its function in transporting oxygen and carbon dioxide. You are given with 3 marks. So you have to talk about binding of oxygen, binding of carbon dioxide with hemoglobin, and why hemoglobin is a suitable way of transporting. So you have to address how hemoglobin is interacting with the polar environment of plasma. Okay, already in this session, we addressed how oxygen is binding. Oxygen binds with the central iron, and carbon dioxide is binding with the amine groups of the proteins to make carb-aminohemoglobin. Hope you can construct your answer to gain 2 marks. We'll grab the other mark. In hemoglobin, I told you that there is a protein component and it is a globular protein, which is folded in a way that polar parts are remaining in the periphery, in the outside, or polar parts are outwards in the structure. So these polar parts are interacting with water or the polar environment of in the red blood cells and get well incorporated or well dissolved in the medium to be a good mean of transportation.